Neurons communicate by passing action potentials from one neuron to the next. So when one neuron generates an action potential, that action potential is going to be generated at the axon hillock, and it's then going to be spread or propagated all the way along that axon to the axon terminal. The axon terminal of one neuron is going to come into very close contact with the cell body or the dendrites of a second neuron. This is where that action potential is going to be passed from the first neuron to the second neuron. So there's going to be this synapse, or a point of chemical transmission, or a point of communication between those two neurons. This occurs between the axon terminal of one neuron and the cell body or the dendrites of a second neuron. This is where that action potential is going to be passed from the first neuron to the second neuron in the form of a graded potential. A graded potential on that second neuron could then lead to an action potential on that second neuron. So the action potential from the first neuron is going to be passed to the second neuron in the form of a graded potential. Graded potentials spread along the dendrites in the cell body towards the axon hillock and can then generate an action potential at the axon hillock of that second neuron. So once again, a synapse is going to be the point of chemical transmission, or the point of communication between two neurons. So this is really where those two neurons are passing the action potentials or nerve impulses from one neuron to the next. For the most part, a synapse is going to occur between the axon of one neuron and the cell body or the dendrites of a second neuron. So a synapse between the axon of one neuron and the cell body of a second neuron is known as an axosomatic synapse. So an axosomatic synapse is a point of chemical transmission or communication between the axon of one neuron and the cell body or soma of a second neuron. Synapses can also occur between the axon of one neuron and the dendrites of a second neuron. This would be known as an axodendritic synapse. So an axodendritic synapse is a point of chemical transmission or communication between the axon of one neuron and the dendrites of a second neuron. Once again, the first neuron is trying to pass its action potential to the second neuron. It produces a graded potential on the dendrites or the cell body of that second neuron. Graded potentials are going to spread towards the axon hillock and then potentially generate an action potential at that axon hillock that could then be spread down the axon of the second neuron. Less common, we see synapses occurring between two axons. So in rare scenarios, you might see one axon synapsing with a second axon. This is very rare. It's much more common for us to see synapses occurring between the axons and cell bodies and the axons and the dendrites. So remember that a neuron has an axon that can branch a number of times. So neurons always have that long process called an axon, and axons can branch. Each axonal branch can then synapse on a different neuron. So one neuron can actually synapse or communicate with many neurons at one time. Additionally, many different neurons can actually synapse on one single neuron. So we can see that right here. So in this diagram here, you can clearly see that 1, 2, 3 neurons are synapsing on this one single neuron. So one neuron can synapse on many neurons, That's because the axons of a single neuron can branch a number of times. Additionally, many different neurons can synapse on a single neuron. A synapse is structurally similar to the neuromuscular junction. However, it's going to be functionally different. One of the major differences between the neuromuscular junction and a neuronal synapse is that at the neuromuscular junction, we always get that muscle to threshold, or in other words, an action potential is always generated on that muscle. This doesn't always occur at a neuronal synapse. So at a neuronal synapse, we don't always have an action potential resulting in response to that first neuron passing its action potential to the second. So we're going to talk a little bit more about why that is later on in this lecture. But for right now, just know that a synapse is very similar to the neuromuscular junction. It's functionally different because at the neuromuscular junction, we are always getting that muscle to threshold. So in other words, when a neuron stimulates a skeletal muscle cell, it's always going to produce an action potential on that muscle cell. That is not always the case at a neuronal synapse. So when one neuron is trying to pass its action potential to the second neuron, that second neuron does not always produce an action potential. That's because we don't always get that second neuron to threshold, which is about negative 55 millivolts. So we're going to walk through the sequence of events occurring at a neuronal synapse. These events should look very, very similar to the events at the neuromuscular junction. So you'll notice a lot of similarities as we're working through these different steps at a neuronal synapse. So in this diagram, we have one neuron shown here. This neuron is known as the presynaptic neuron. So the presynaptic neuron is going to have an action potential coming down its axon and arriving at the axon terminal. This presynaptic neuron is trying to pass its action potential to the second neuron, known as the postsynaptic neuron. So the postsynaptic neuron is trying to receive that action potential or receive that nerve impulse. You'll notice that this is an axosomatic synapse because it's a synapse or a point of chemical transmission between the axon of one neuron and the cell body or soma of a second neuron. So what's being shown here is an axosomatic synapse because it's between the axon and the soma or cell body. So when an action potential comes down this axon, it's going to arrive at the axon terminal. Remember that an action potential is a change in membrane potential or a change in voltage. The axon terminal has voltage-gated calcium channels that are present and are going to respond to the arrival of that action potential. So the action potential arrives at the axon terminal. It triggers the opening of voltage-gated calcium channels. That causes calcium to flood the axon terminal. As intracellular calcium levels rise in the axon terminal, that triggers the fusel of these synaptic vesicles containing a neurotransmitter with the plasma membrane of the axon terminal. So these synaptic vesicles are going to contain the neurotransmitter. They're going to fuse with the plasma membrane of the axon terminal, releasing that neurotransmitter by exocytosis into the synaptic cleft. The synaptic cleft is just the space in between that presynaptic neuron and postsynaptic neuron. So an action potential comes down the axon, arrives at the axon terminal, triggers the opening of voltage-gated calcium channels, causing calcium to flood the axon terminal. A rise in intracellular calcium triggers the fusel of these synaptic vesicles with this plasma membrane and the release of the neurotransmitter into the synaptic cleft. The neurotransmitter is then going to diffuse across that synaptic cleft, and it's going to bind to its receptors on that postsynaptic neuron membrane. So action potential arrives at the axon terminal. That triggers the opening of voltage-gated calcium channels. Calcium enters that axon terminal, triggering synaptic vesicles to fuse with the membrane, releasing the neurotransmitter into the synaptic cleft by exocytosis. That neurotransmitter is then going to diffuse across the synaptic cleft and bind to its receptors on the postsynaptic neuron. The receptors on the postsynaptic neuron are known as chemically gated ion channels or ligand-gated ion channels. So when these neurotransmitters bind to their receptors, it then opens these channels, allowing for ions to flow into or out of that postsynaptic neuron. So we can see that right here. When our neurotransmitter binds to its receptors are chemically gated ion channels or ligand-gated ion channels. So when the neurotransmitter binds, it opens these channels, allowing for ions to move into or out of that postsynaptic neuron. Remember that ions are charged molecules. So if you have charged molecules moving in and out of that postsynaptic neuron, you're going to have a change in voltage across that plasma membrane, and that change in voltage is going to be known as a graded potential. So the binding of a neurotransmitter opens ion channels on that postsynaptic membrane, resulting in a graded potential. Synapses can be excitatory or inhibitory. At an excitatory synapse, the postsynaptic membrane is becoming closer to threshold. So that postsynaptic membrane is actually becoming more positive. At an excitatory synapse, when that neurotransmitter binds to its receptors on the postsynaptic neuron, when these receptors or these channels open, it's then going to cause an influx of positively charged ions. So at an excitatory synapse, we're bringing that postsynaptic membrane closer to threshold or closer to -55. So we're going from -70 resting membrane potential to -55 threshold. So we're getting more positive. So that means there's likely going to be an influx of positively charged ions into that postsynaptic neuron. That's helping to bring that postsynaptic neuron to threshold, hopefully allowing then for an action potential to be generated on that second neuron or on that postsynaptic neuron. At an inhibitory synapse, the postsynaptic membrane goes away from threshold, so the cell is actually getting more negative. So if it's an inhibitory synapse, that postsynaptic membrane is going to go from -70 to maybe -90. So the cell is becoming more negative and moving farther and farther away from threshold in order to prevent an action potential from occurring. So at an inhibitory synapse, when the neurotransmitters come across and bind to these receptors, when these channels open, it's likely causing an efflux of positively charged ions, or a loss of positively charged ions. That's what's making that postsynaptic neuron more negative. It's going to be the loss of those positively charged ions. So synapses can be excitatory or inhibitory. At an excitatory synapse, we're bringing that postsynaptic neuron closer to threshold in hopes that it will elicit an action potential. So the cell membrane is becoming more positive or getting closer to that -55 threshold because of an influx of positively charged ions. At an inhibitory synapse, we're trying to inhibit an action potential from occurring on that postsynaptic neuron. So we're bringing that postsynaptic neuron farther away from threshold or making that cell become more negative. This is caused by an efflux of positively charged ions or a loss of positively charged ions. So binding of a neurotransmitter is going to open our ion channels, producing either excitatory or inhibitory graded potentials. The neurotransmitter's effects are going to be terminated by reuptake through transport proteins, enzymatic degradation, or diffusal away from the synapse. So the neurotransmitter's effects are going to be terminated because the neurotransmitter is going to be removed from the synaptic cleft. There are 3 ways the neurotransmitter could be removed. It could diffuse away. It could be degraded by an enzyme in the synaptic cleft, or it could be recycled back into that presynaptic neuron. So there could be reuptake of that neurotransmitter into that presynaptic neuron through these transport proteins. So the removal of the neurotransmitter can occur in 3 different ways. It could simply diffuse away. It could be enzymatically degraded. We've seen one example of this at the neuromuscular junction. If you remember, we said that the enzyme acetylcholinesterase is present and can degrade the neurotransmitter acetylcholine. So enzymatic degradation could remove that neurotransmitter, or that neurotransmitter could be recycled back into that presynaptic neuron by reuptake through transport proteins. So the neurotransmitter's effects are terminated by reuptake via transport proteins, enzymatic degradation, or diffusion away from the synaptic cleft. In any of these cases, that neurotransmitter is removed from the synaptic cleft, so its effects are terminated. Nerve impulses can travel extremely quickly down an axon of that presynaptic neuron. So nerve impulses or action potentials can travel extremely fast, about 150 meters per second or about 300 miles per hour. This is extremely fast in comparison to how quickly a chemical transmission occurs at a synapse. So an axon coming down that presynaptic neuron is going to occur much faster than the neurotransmitter being released from the presynaptic neuron, diffusing across that synaptic cleft, and binding to that postsynaptic neuron, producing that graded potential. So we have something called the synaptic delay. The synaptic delay just reflects the time required for that neurotransmitter to be released, diffuse across the synaptic cleft, and bind to its receptors. This just shows that transmission across a chemical synapse is extremely slow in comparison to how quickly that nerve impulse can actually travel down an axon. So we call it the rate-limiting step in a neural pathway. The synaptic delay takes about 0.3 to 0.5 milliseconds. So in other words, that's about how long this process takes for neurotransmitters to be released, move across the synaptic cleft, and bind to their receptors, producing those graded potentials. This is important because it helps explain why neural pathways that involve just 1 or 2 neurons are going to occur much quicker than neural pathways that occur require 5 or 6 neurons. So if you have a neural pathway that just has 2 neurons, you only have one synapse between those 2 neurons. So you only have one spot where we have a synaptic delay. Versus if you have 4 neurons or 5 neurons synapsing in one neural pathway, you're then going to have 4 or 5 spots where you're going to have a synaptic delay occurring. So transmission is going to occur much quicker in neural pathways that have fewer neurons in comparison to neural pathways that have many neurons, because there's fewer synapses and therefore fewer synaptic delays.